Yes, welcome back to our another video tutorial. This is Rajesh Palakurti. Welcome to RS Biology tutorial. Now in this session we have to discuss about structure of double-stranded polynucleotide chain in the chapter of molecular basis of inheritance. Okay, we know about that every DNA is made up of, generally each and every DNA is made up of two strands. Each and every strand is made up of many nucleotides. That's why the strands are called as polynucleotide chains here. Okay. In this session, we are going to discuss about what is the structure of two strands of the DNA. They are nothing but polynucleotide chains here. Okay. Suppose this is the one strand of DNA. This is the another strand of DNA. Each and every DNA is made up of two strands, and every strand is made up of many nucleotides. That's why they are called as polynucleotide chains. Suppose this is the one polynucleotide chain, and this one also another polynucleotide chain here. Okay. In this session, we want to know which type of bonds are present in the between the different type of nucleotides, and within the nucleotides which type of bonds are present, how the new DNA strand will be synthesized, what is the direction of growth of the new DNA strand. Everything we will discuss in this session. Okay. See here. Suppose here in this strand, suppose it is one nucleotide. Suppose it is pentose sugar. A means adenine, nitrogen base, and P means phosphate. Suppose here from here to here it is one nucleotide. We know that nucleotide consists of pentose sugar, nitrogen base, and phosphate. One nucleotide. Suppose in the same way it is also one nucleotide. Nitrogen base, pentose sugar, and phosphate. Another nucleotide. In the same way, nitrogen base, pentose sugar, and phosphate. It is also one nucleotide. Here also C for nitrogen base, cytosine, nitrogen base, pentose sugar, and phosphate. It is another nucleotide. Total how many nucleotides there in this chain? 4 nucleotides are there. Suppose it is first nucleotide of this chain, it is second nucleotide of this chain, and this is the third nucleotide, and this one is the fourth nucleotide. Suppose in this chain, this is the first nucleotide, this is the last nucleotide. Okay, we know that each and every nucleotide is made up of nitrogen base, sugar, phosphate, nitrogen base, sugar, phosphate, nitrogen base, sugar, phosphate. Here also nitrogen base, sugar, phosphate. We know that. The same way in this strand also different kind of nucleotides are formed here. Suppose here it is nitrogen base, pentose sugar, and phosphate. It is one nucleotide. Same way C, nitrogen base, pentose sugar, and phosphate. It is another nucleotide. Next nitrogen base, pentose sugar, phosphate. Another nucleotide. They are also same. Nitrogen base, pentose sugar, phosphate. Here also 4 nucleotides. But here now it is first nucleotide. This one is the second nucleotide. This one is the third nucleotide, and this one is the fourth nucleotide. In the case of lower polynucleotide chain, the first nucleotide should be this one, and the last nucleotide should be this one here. Okay. What is the reason behind this? We will discuss later. Now see here. In this picture, this DNA strand is made up of how many nucleotides? 4 nucleotides. And this polynucleotide chain also made up of 4 nucleotides. We know that, right? Okay. What about the bonds? Which type of bonds are present in the double-stranded polynucleotide chain? There is maybe 4 type of bonds are present here. 4 type of bonds. The first one is, see, in the case of this nucleotide, this is phosphoester bond. Already we know what is the phosphoester linkage. The linkage present between the pentose sugar and phosphate. This linkage is called as phosphoester linkage. Okay, the linkage takes place between the pentose sugar and phosphate is called as phosphoester linkage, right? And what is N-glycosidic linkage? The linkage takes place between C. C means what here? It is one type of nitrogen bases. The linkage takes place between nitrogen base and pentose sugar is called as N-glycosidic linkage. Okay. Next, hydrogen bonds. Very, very important. Hydrogen bond. Yes. See here, this strand having some nitrogen bases and this strand also having some nitrogen bases. Okay. What is the mode of bonding between the different nitrogen bases of two opposite strands? Always one purine can bind with one pyrimidine. As usually one pyrimidine can bind with one purine. There one rule is there. Always adenine bind with thymine. Adenine of one polynucleotide chain is bind with thymine of its opposite polynucleotide chain. The same way, suppose in upper chain there is thymine, in lower chain there should be adenine. Here the upper chain there is guanine, in lower chain there should be cytosine. Here cytosine, here guanine. Always adenine in DNA, especially in double-stranded DNA, always a purine, adenine combined with a pyrimidine, thymine. Okay, adenine combined with thymine. As usually, thymine combined with adenine. The same way, guanine combined with cytosine, and cytosine also combined with guanine. This is the one rule. This rule is prepared by another scientist. We will discuss later about that. Just remember here, always between A and T, there is two hydrogen bonds. Between adenine and thymine, how many hydrogen bonds are present? Two hydrogen bonds are present here. And between G and C, there is three hydrogen bonds. This is the rule, remember always. Okay, right. Up to now we discussed three types of the bonds here. One is phosphoester linkages and N-glycosidic linkages and hydrogen bonds here. But what about phosphodiester linkages? Phosphodiester. We know that phosphodiester linkages are formed between two successive nucleotides. Okay, suppose it is one nucleotide, it is next nucleotide. Between this bond is there, no? This linkage. Yes, this linkage is called as phosphodiester linkage. Suppose see in this case, here phosphate is there, here one ester bond, here one ester bond. Phosphate having two ester bond. Either side of the phosphate there is ester bond. That's why it is called as di. Why we are calling here di? When phosphate having two ester bonds of successive nucleotides, that's why it is called as what? Phosphodiester linkages. Phosphodiester linkages. Okay, right. Once again I am repeating here, mode of bonding, N-glycosidic linkages formed between pentose sugar and nitrogen base. What is the location of N-glycosidic linkage? It is between pentose sugar and nitrogen base. And what is the location of phosphoester bond? It is formed between nucleoside and phosphate. Nucleoside and phosphate. And where the hydrogen bonds are formed? Formed between nitrogen bases of two opposite strands. Nitrogen bases of two opposite strands. Suppose these are the nitrogen bases of two opposite strands. Between these two nitrogen bases, which type of bonds are present? Hydrogen bonds are present. Between A and T, two hydrogen bonds. Between G and C, three hydrogen bonds. Okay. Yeah, and up to now we discussed about four type of the bonding in a structure of double-stranded polynucleotide chain here. Okay. Now suppose what is these arrow marks? These arrow marks are opposite together. That means the direction of growth of new DNA strand always 5 to 3 direction here. 5 to 3 direction. What is the 5 to 3? Here, this end of this polynucleotide chain is termed as third end and this end is known as fifth end or fifth prime, third prime. Why we are calling like this? Suppose here fifth prime, here third prime. Why we are calling like this here? Suppose see. Suppose it is first nucleotide. First nucleotide. Before this nucleotide, there is no any nucleotide. So it is only first one. It is beginning nucleotide. First one, initiative nucleotide. That's why here which carbon is free? Fifth carbon is free. Fifth carbon. You must remember before that always the every successive nucleotides are bind together in 3 to 5 direction. See here 3 to 5, 3 to 5, 3 to 5 direction. That means always phosphodiester linkages are formed in which direction? 3 to 5. That means phosphodiester bonds are phosphodiester linkages are formed between third carbon of first nucleotide with fifth carbon of next nucleotide. See here, for third carbon of this nucleotide with the fifth carbon of this nucleotide. Third carbon, fifth carbon. That's why phosphodiester linkages are called as what? 3,5 phosphodiester linkages. Okay. See, in this case also same. Suppose phosphodiester, this is the phosphodiester linkage. Phosphodiester linkage formed between third carbon of one nucleotide with the fifth carbon of next nucleotide. 3 to 5 direction always. Okay. Whenever the phosphodiester bonds are formed in 3 to 5 direction, always the direction of growth of every new strand of DNA should be 5 to 3 direction. 5 to 3. Suppose why it is called fifth end? When fifth carbon is free here, fifth carbon is not attached with any other nucleotide here. Fifth carbon is free. That's why this end is called as what? Fifth end or fifth prime. Whenever here also, suppose it is last nucleotide. In this last nucleotide, the third carbon is free. Why? Because it is only last one. There is no another nucleotide to bind with it. That's why here third carbon is free. That's why this end is called as what? Third end. Okay. In this way, in the same way, here fifth carbon is free. That's why it is called as fifth end. Here third carbon is free. That's why it is called as third end. You always must remember, in the case of upper chain, this is the first nucleotide. This is the last nucleotide. In the case of lower chain, this is the first nucleotide. This is the last nucleotide. Okay. Always the growth direction of every new DNA strand or any RNA strand always 5 to 3 direction. Suppose what is the growth direction of upper chain? 5 to 3 direction. Here also here 5, here 3 no. 5 to 3 direction. 5 to 3 direction. Okay. This condition is called as anti-parallel condition. What about anti-parallel? We will discuss later. Now simply see here. Every DNA strand growth direction always 5 to 3. 5 to 3. If anyone asks what is the reason behind that, always the new DNA strand grow 5 to 3 direction. What is the answer here? Yes. Due to formation of 3,5 phosphodiester bond. Due to formation of 3,5 phosphodiester bond, always the new strand direction of growth is 5 to 3. 5 to 3. That is your answer. Okay. Why the growth direction of new strand is 5 to 3 always? This is the question. What should be the answer here? Due to the formation of 3 to 5 phosphodiester linkages. 3 to 5 phosphodiester linkages. Due to the formation of 3 to 5 phosphodiester linkages, always the new strand direction of growth is 5 to 3. Here also same. Phosphodiester linkages direction is 3 to 5. That's why new strand growth direction is 5 to 3. 5 to 3. Okay. Yes. This is the structure of polynucleotide chains of double-stranded DNA. Double-stranded DNA. Okay. We know that it is NCERT diagram as it is. We are trying to decoding the NCERT lines. I hope you understand the concept. Okay. If you got any doubt, you can say in the comment sections. And okay, we will meet in the next video. I hope you understand the concept. Thank you.